My name is Igor Polikarpov. I'm a full professor of the São Carlos Institute of Physics at the University of São Paulo. And my talk is one of the talks at technical session 19 of BBEST 2021. In my presentation, I mostly will focus on thermocellulomyces thermophilus to the main LPMO 9H. Thermocellulomyces thermophilus is also known as Methylophthora thermophila, and this is one of the commercially relevant fungal expressions. So we looked at the light stimulation, low resolution science model with cellulases of the LPMO. And what is LPMO? LPMO stands for lytic polysaccharide monooxygenases, which are monocopper enzymes that are capable of oxidatively cleaving recalcitrant polysaccharides, for example, cellulose. And by doing so, they have important biotechnological applications in, for example, second generation ethanol and cellulosic ethanol production. The copper atom of LPMOs is coordinated by so-called histidine braids by the side chains of two histidine residues. These enzymes catalyze both monooxygenase and peroxygenase reactions, and these reactions result in oxidative cleavage of polysaccharide chain. During this process, oxygen atom will be inserted in C1 or C4 position of beta-1,4 linkage of cellulose, for example. LPMOs are classified in several different families in CAZy database, namely A, 9, 10, 11, 13, 14, 15, and 16, and the LPMO we were studying from AA9 family. So the enzymes frequently are encountered as single domain enzymes, and they're small, they're very robust and stable, and coordinate copper atom by, as I said, side chains of the residues. One of them is histidine. When we decided to characterize TG-LPMO9H, it had to be produced. So we hydrolyzed expressed it in aspergillus nidium. And when you produce something in fungal expression system, you will check the resulting purifying enzyme for activity, additional activities such as cellulase activities. We used two synthetic substrates, PIPG and PNPC, which are substrates for glycoside hydrolysis, but not of LPMOs. Unfortunately, we detected quite strong activity against PNPG, which indicated the presence of glucosinase. Well, we had to do something about it, and what we did, we decided to heat up and cool down LPMO, basically hoping that the enzyme will nature after thermal penetration, and that exactly what happened. We increased the temperature for up to 85 degrees and supplied copper atoms, and with that activity of LPMO was and regional activity, whereas activity in PNPG was gone. Well, then we looked at the substrate specificity of this enzyme, and basically, to be able to do so, we used a so-called futile cycle of LPMO reactions, where LPMO in the absence of substrate is known to produce hydrogen peroxide. If one would supply the enzyme, the substrate to the enzyme, then concentration of hydrogen peroxide will go down. That's exactly what happens. Either the hydrogen peroxide production by LPMO at the absence of substrate, and here in the presence of phosphate. Considerable decrease in the hydrogen peroxide production was also observed for oxaloglucan, which is presumably a substrate for LPMO. Well, since the bad substrate was passed, we used the substrate for enzymatic activity as well as other cellulose substrates like avicell. Well, first of all, we looked at the pH dependence of peroxygenase activity of LPMO, and it's actually quite strongly pH dependent. The highest activity is actually observed at alkaline pHs, but enzyme is more stable in neutral and acid pHs. We use these pHs in further studies. And while acting at cellulose, the enzyme activated by aspartate gallate is capable of oxidizing between 4 bonds both at C1 and C4 positions. Well, we next looked at the light activation of the LPMO, and for that we looked at the activity of LPMO in the presence of chlorophyllene and ascorbic acid in the dark and under light. And one can see that when we shine light at the chlorophyllene and it activates LPMO, LPMO is producing way more products than at the absence of light. It means that it is, in fact, light activated. Well, we next looked at the synergy with different endoglucanases, and for that we have chosen 4 different endoglucanases from 4 different GH family, GH5, 7, 12, and 45, and clearly there is a huge synergy with GH7. LPM also activates GH5, 12, and 45, but not to the degree that it activates GH7 and the glutamate activity. Well, LPMO is capable of activating endoglucanates. We said, well, let's have a look at the cocktail, enzymatic cocktail, and we mixed it with celluloplast. And indeed, when this mixture was acting at 2 different and 3 differently producing biomass, sugar in the gas, it clearly was more active than the same mixture that the effect was particularly strong for steam-loaded sugar in the gas. We finally looked at the low-resolution model of LPMO. And indeed, small angle scattering studies, the 2-domain LPMO was capable of revealing the low-resolution angle of the LPMO. You see, no structural information was known for 2-domain LPMOs before our study, and simply because it's very difficult, if possible at all, to crystallize 2-domain LPMOs. Well, one can study them in solution, and indeed, the obtained envelope clearly shows 2 different lobes. The bigger one, which one can have quite successfully fit high resolution knowledge model of the catalytic domain, and the small lobe in which one can fit CBM model. SACS data also allowed to retrieve the conformation of the lignocellulosic, and one can see a huge long lignocellulosic that coordinates relative position or orientation of these two models in such a way that the plane phase of CBM looked at the same direction as the plane phase of the catalytic domain. So one can imagine that the cellulose plane in which LPMO actually can be positioned here, and the enzyme is looking downward on the cellulose plane. Well, these are the results of our studies of TTL-PMO. I will summarize what we have concluded or found out. TTL-PMO9H is a two-domain LPMO with C1, C4, reduced selectivity. Contaminants of LPMO fungal recombinant production can be eliminated by thermal treatment, and that's in general procedure can be applied to any thermostable LPMO which is capable of thermal renaturation. The enzyme can be activated by light in the presence of chlorophyllin and ascorbic acid. TTL-PMO9H shows considerable synergy with J7 endoglucanases, but not with endoglucanases from J5 to 45 families. LPMO also, these LPMO also can boost commercial enzymatic cocktail. This effect depends on environment treatment. Finally, we retrieved low resolution SARS model, which shows tadpole-like envelope with a plane aligned CBM and catalytic domains joined by a long linker peptide. Well, I would like to thank all of you for your attention.
My name is Pedro de Oliva Neto. I am professor of Unesp University, São Paulo, in São Paulo State. And I would like to present our work with my student Bruno Scaramboni, new technological opportunities of feedstock to improve the sugar and ethanol industry. I'd like to thank the organizing committee for these opportunities. And why this subject? Why feedstocks? New feedstocks. Sugarcane and starch, for example, is enough enough for the ethanol production in Brazil? Yes, but if we can improve the production of ethanol with other kind of products, we can use more lands for food, for example, and using some new feedstocks for the increase of ethanol. I think it's necessary in Brazil. So in this case, we are using as an example of this way, sisal waste, cassava bagasse, and food waste, and we will show our results. Sisal waste is a mucilage when we extract this fiber, sisal fiber, from this plant. They cut these leaves in the field, and the fiber is produced, but 95% of these leaves is wasted. So this is the region, the idea of to produce sugars and other bioproducts like these patents that we have at Unesp from mucilage. Okay, about 85.7% is full wet mucilage, and we when we do it, we extract the sugars. We have about 1.5 to 3% of sugars in leaves. So this is the mucilage. And in the juice, when we analyze the juice, we have 5 to 12% of sugars. So if we combine the sugars from the juice, from the mucilage, the fiber mucilage without the juice, we have 190 million liters of ethanol. It's possible to generate, to produce by second generation technology, this sum of ethanol from these plants in a semi-arid area, in a very poor area, helping people that need these technologies. The second option is cassava bagasse. Why cassava bagasse? Because we have 3.5 million tons of in dried basis of cassava bagasse, a waste from a solid waste, semi-solid waste from the extraction of starch. So using cassava, the plant cassava, mandioca in Portuguese. And here we have our methodology to extract the starch and the glucose in the final technology. We have glucose as final product. When you use, for example, here we use wastes or white or other products, in general wastes, for the production of fungal amylases by Rhizopus oligosporus. After we use the fungal amylase with cellulase or not, and produce glucose in the hydrolysate, and after we use the hydrolysate, fermenting with Saccharomyces cerevisiae and producing ethanol. This is our strategy. And here we have a patent and a publication about this technology with more details. We use a combination of salts to improve this production. It's very important in a solid state fermentation using Rhizopus oligosporus and wet bran or wastes too. And here we studied the hydrolysis of cassava bagasse using amylases and cellulases in different concentrations. We got optimization of this process with these equations, and with this formula or combination of 15 units of amylase and 5 units FPU of cellulase, you can convert 52% of cassava bagasse into fermentable sugars in 36 hours. So it's very interesting, considering cassava bagasse costs about 30 reais 1 ton, with 85% of unit moisture. So it's very economic. Here we have 15% of concentration of this substrate in our hydrolysis in fermenter, and with that model of 15 amylase and 5 cellulase, and we got 45% of conversion in glucose with this high concentration of cassava bagasse. So very, very interesting, thinking in an industrial scale. We can see here the attack of cellulase and amylases, the combination in a fiber, in the cassava bagasse. And we have here, with this combination of enzymes, an ethanol yields efficiency of 78%, and the production of about 254 liters of ethanol per ton of cassava bagasse. It's a huge number. Here we can see the kinetics of our amylase produced in our conditions, competing with commercial glucoamylase and very expensive glucoamylase. Our is very, very economic. So in the same, we have the same design of this kinetics. And here we have 43%, about 43% of hydrolysis of cassava bagasse, and here only pure starch, right? And we can combine, compare the cost of feedstock, cassava bagasse, and sugarcane. If we compare only the feedstock, it's a half part of the cost of sugarcane. So it's economical too. And we can improve the profit of technology using cassava bagasse. Other option of feedstock is food waste. Food waste is produced in the world about 1.3 billion tons. So it's a big number. And why not use this product to produce ethanol, biofuels in general? For example, ethanol, if we have about 50%, 48% of carbohydrates in our numbers, and lipids about 16%. So we can produce biodiesel and bioethanol together by the same feedstock when we hydrolyze them. And we can use food waste for the production of our amylase too. We have here a good numbers, 170 units per gram of amylase and 270 units per gram of protease together in the same extract, using food waste in a half parts, and cassava bagasse and wet bran, the others, the other 50%. So it's possible to use, and in an economical process, considering food waste, we don't have cost for food waste. The opposite, the food waste is spend money to solve this problem for the cities in Brazil and in the world. So we are in an economical process and helping in the environment too. And here we have some other results, about 36.4% of conversion, only using amylase and food waste. If you consider the three parts, three feedstocks in only this example, we have 5.5 billion liters of ethanol that we can produce, and it represents 15.5% of the last crop, 35.6 billion liters. So it's not low, it's a good number. We can improve the ethanol production only with these three options. We have more. So it's important to study these new options of feedstock and include in our technology of ethanol production in Brazil. This is our conclusions. We can produce ethanol by other feedstocks considering a good results with food waste, sisal waste, and cassava bagasse. And the use of our enzyme glucoamylase from Rhizopus oligosporus was very efficient, and we can use this enzyme in a scale up, considering our results. Thank you very much for this opportunity. My students, Bruna, participate in this work, and others students and Professor Fernandes Nunes and others that in the past helping us about these results. Thank you very much.
Hi everyone, I'm Taisa Dinamarco. I'm a professor at Chemistry Department at São Paulo University in Ribeirão Preto. It's a pleasure for me today to speak at the BBEST virtual conference. I'd like to thank our organizers, especially Professor Igor Polikarpov, for the invitation. The demand for energy has increased continuously worldwide, which has raised concerns about the sustainability and the development of advanced in renewable and sustainable source of energy. In this context, bio-based economy or bioeconomy refers to economic activity involving the sustainable production and use of biological resources, processes, and principles to provide products and services in all economic sectors. The bioproducts made from biomass are characterized by a variety of properties, compositions, and processes, as well as different benefits and risks, are developed in order to reduce dependence on fossil fuels and non-renewable resources. To achieve this, the key is to develop new biorefinery technologies to sustainably transform renewable natural resources into bio-based products, materials, and fuels. Biomass is composed of three biopolymers, such as cellulose, and lignin, which confers a recalcitrant structure to these plant cell walls. To deconstruct plant cell walls and release soluble sugars, three steps are involved: physical and chemical pretreatment, and sequential enzymatic hydrolysis, and final yeast fermentation to produce bioethanol. Due to the intrinsic recalcitrance of lignocellulose, it becomes essential to explore optimal biomass process technology for efficient bioethanol production, such as better hydrolytic enzymes. Our group has been working in identify and characterize new hydrolytic enzymes that can increase the capacity of enzymatic cocktails and decrease the cost involved in this process. So, our main objective is to characterize new thermostable enzymes that can be supplemented in enzymatic cocktails. In order to identify potential new genes and proteins involved in SEB, sugarcane explosive bagasse breakdown, we analyze the transcriptome and secretome of Aspergillus fumigatus in the presence of sugarcane bagasse. First of all, we analyze the activity of the enzymes secreted in the medium, and we detect this activity after 3 days of culture, except xylanase. No enzymes activity were detected prior to 24 hours. To get more insight in the gene expression, transcriptome was performed by RNA sequencing to compare the expression profile of Aspergillus fumigatus when it was grown on sugarcane explosive bagasse or fructose as a carbon source. We found 197% different expressed case enzymes genes, which represent 35% of all case enzymes encoded in Aspergillus fumigatus genome. As you can see here, these genes had a different expression profile. Some of them were upregulated and others were downregulated. As you can see in this table, some of these enzymes were characterized and were upregulated or downregulated. As you can see here, AN9 was upregulated in this situation and was the first time described in Aspergillus fumigatus transcriptome. Once the transcriptome was characterized, we analyzed the secreted protein by SDS page and MS-MS. We detected 128 secreted protein in sugarcane exploded bagasse supernatant being 101 exclusively for the biomass. This graph represents all the proteins detected in cell culture, and many CAZymes were observed in these proteins. Crossing these results, we could see that some of these enzymes were also detected in transcriptome and secretome analysis of Aspergillus fumigatus, and many of them were also upregulated and present in the secretome. So we understand that these enzymes play an important role in biomass degradation. So we started to characterize some of them. The first enzyme I want to show here is an endoglucanase that was expressed in two different systems as E. coli and PIPG pastures. All the biochemical parameters were determined as optimal temperature at 55 degrees, pH 5, thermostability in different temperatures, and a range of pH. These enzymes were highly stable at 55 degrees up to 72 hours of incubation. The effects of metal ions and chemical substrate were also analyzed for a barrier characterization, and cinetic parameters were also analyzed and showed a good activity in both substrates, CMC and beta-glucan. To analyze the performance in biomass degradation, we supplemented the cellulase cocktail cell class with 10 micrograms of these enzymes and incubated at 55 degrees for up to 72 hours. Interesting, these enzymes showed a high degree of synergy with the cocktail deconstruction, except in barley bagasse. Two other enzymes were characterized by our group are LPNO, AN9, and cellulose CYL6A. We characterized all the biochemical parameters of both enzymes, as we can see here, including the effect of substrate in enzymes activity. We can see that both enzymes were tolerant to glucose up to 250 millimolar, keeping around 90% of residual activities. At 100 millimolar of cellobiose, it was inhibiting 50% of cellulose activity and 8% of LPNO. But the most important result I would like to show is the synergistic action of both enzymes in CM cellulose. The reactions were performed at different enzymes proportions at different incubation time. Surprisingly, we observed an inhibitory effect when both enzymes were associated. We also investigated the synergistic effect of both enzymes on cell class cellulase cocktail. Hydrolysis increased over the time, and compared to cocktail alone, the addition of LPNO or cellulose increased the reducing sugar released, suggesting that they boost this enzymatic cocktail. No inhibitory effect were observed, probably because LPNO in this situation were acting together with other enzymes present in cell class cocktail. Finally, we associated the three enzymes previously characterized by our group. It showed a positive effect when associated with the endoglucanase, but again, we observed an inhibitory effect of cellulose and LPNO. To evaluate how these enzymes acted on lignocellulose biomass, we analyzed the association complex biomass such as seb, corn cob, and rice straw. We observed an increase in release of reducing sugar in seb and corn cob, but not in rice straw. Besides that, LPNO had a negative effect when we added in cell class cocktail on corn cob and rice straw biomass. The divergent results point to the substrate dependency or specifically activity of these enzymes in this substrate. And here, when we compare the association of the three enzymes in biomass, when comparing the endoglucanase alone, we observed an increase in reducing sugar, and different proportions depend on the biomass. LPMO association had no effect or a little impact hydrolysis after 48 hours in the three substrates. Finally, we characterize a xylanase. All the parameters were determined, the best conditions of activity were consistent with the other xylanase published in the literature, but only few numbers of enzymes showed optimal activity in temperatures as high as 8 degrees. We added increased concentrations of monosaccharides as glucose and xylose to analyze the inhibitory effect of the sugars, and the enzymes remained stable up to 250 millimolar of xylose and glucose, and the ethanol decreased the activity of these enzymes when we increased the concentration of ethanol more than 1%. The effect of synergist action between xylanase and cell class on sugarcane exploded bagasse or sugarcane bagasse without lignin, we observe that the a synergistic effect that boosts the activity in order to 160% in sugarcane exploded bagasse, or more than 300% when considered a biomass without lignin. This data also reinforced the limits imposed by lignin on the recovery of sugars from biomass. It evidenced by the magnitude of the difference between the scales of the graph. We analyzed the synergist action between xylanase and the characterized LPMO, but no synergistic action were observed. So the main conclusions are: novel enzymes characterize well the saccharification of different biomass. The different synergistic effects are important to understand the actions of LPMOs with cellulase and would help to design new commercial enzymatic cocktails. Substrate composition affects the synergism with cellulase. I'd like to thank you, all the supported by FAPESP, CNPq, CAZymes, BON, and the Chemistry Department, São Paulo University. This is our team. I'd like to thank you for your attention, and I'm open for questions. Thank you.
Hello, everyone. My name is João Luís Carvalho, and I am a researcher at Brazilian Biorenewables National Lab. And today the title of my presentation is Understanding the Pros and Cons of Crop Residues Removal for Bioenergy Production on Soil Greenhouse Gas Emissions. So I will start with a brief introduction about the sugarcane harvest in Brazil. Until 15 years ago, most part of the sugarcane harvest was conducted under manual and burned harvest system. But this system changed a lot, and in 2020, for example, 97% of the area under sugarcane production in South Central Brazil was harvested without burning and under mechanized conditions. So in this scenario, where the sugarcane was harvested without burning, we have the presence of a large amount of the straw on soil surface. On average, we can observe that the maintenance of a large amount of straw on soil surface represents several benefits to soil plant systems. But on the other hand, the sugarcane straw represents around 1/3 of the energy contained of sugarcane plant. The other 2/3 is represented by sugarcane bagasse and sugarcane juice that is used to produce sugar and ethanol for first generation roots. So the current dilemma that we have here is how much sugarcane straw should be removed from the field without compromise soil health. So what is the best use of the straw? So, but more specifically, if we are talking about our current presentation today, it's the question is, what is the impact of the straw removal on soil greenhouse gas emissions? So based on that, the objectives of these studies, here I am present three objectives. The first one is quantify the impact of the straw removal on soil carbon stocks and N2O emission in areas under different adaptive climatic conditions. So the second one is to derive direct N2O emission factor that represent the regional specific conditions of sugarcane production in Brazil. And finally, this study aimed to calculate the net soil GHG emissions in CO2 equivalent, considering the change in soil carbon stocks and also the N2O emissions under different scenarios of straw removal. So I will start to talk about the methodology that we use in this study. I divide the methodology in two parts, one methodology for soil carbon stocks measurements and the other one for N2O emissions. We conduct 10 field experiments to evaluate the change in soil carbon stocks. These experiments were grouped in two groups, one associated with sand soils, and the second one is associated with clay soils. It's important to mention that the field experiments that we conducted were located in São Paulo state with seven experiments, and 3 experiments were located in Goiás state. So here I am showing the straw removal rates that we use in this study. We use no removal, where 15 megagrams of the straw was maintained on the field per year. Low removal, 10 megagrams of the straw. High removal, 5 megagrams of the straw, and total removal, where all straw was removed from the field. Our field experiments start at after plant cane harvesting, when straw were available in the fields, and we did a soil sampling at before the straw removal treatment establishment, and after 2 years in 4 areas, and after 4 years in 6 areas. Now the methodology associated with N2O emissions. We conduct 4 field experiments to measure N2O emissions. We use the static chamber methodology, and the greenhouse gas measurement was done using a gas chromatography. The gas samples were collected during 2 years, and the results of the emissions were expressed in terms of cumulative emissions and N2O emission factors associated with the application of N fertilizer in different scenarios of straw removal. And we also calculate the N2O emission factor according to this equation here. Let's start to talk a little bit about the results. So in the first moment here, I am present the effect of the straw removal on soil carbon stocks. Here in this bar, we are present the effect until 10 centimeters, and here until 30 centimeters. on average, the lower carbon stocks were observed in total removal scenario. Sometimes the results were different, significant different, sometimes not. Sometimes not. Here, for example, we have sometimes it's not so easy to observe effects of the straw removal in short term, 2 years, for example. But the objective here was analyze all field experiments together. When we analyze all field experiments together, we observe that each ton or megagram of the straw that is maintained on the field result in accumulation of 55 kilograms of carbon per megagram of the straw maintained on the field. When the data were converted in CO2 equivalent, was around 200 kilograms of CO2 equivalent per megagram of straw. This value here will be used in other slides in order to compare the results in terms of CO2 equivalent. And about the N2O emissions. Here I am present just one example of one area during 2 years. So here I am present the data of N2O emission the first years, here in the second year, the same year in two different years. In this graphic, it's possible to see the lower emission, N2O emission was observed in total removal scenario, and the high emission was observed in no removal. In the second year was a little bit different, just in these two treatments differ from the first year, but in average the result is pretty much the same. In this graphic here, I am showing the results of all areas together. What is possible to see here? It's possible to see that the first moment is the value proposed by IPCC that represent then 1% of the nitrogen added in the soil as fertilizer or as a straw is lost by N2O. In our case, here, if you analyze just the asterisk that means the average number, it's possible to see that in all scenarios of straw removal, the value proposed by IPCC is overestimate the direct emission from N fertilizer plus straw. So this is one important information including this graph, and the other one that regardless the scenario of the straw removal, the results observed in Brazil are below the IPCC, and in the other one is there is a tendency of increase in N2O emission according of the amount of the straw that we maintain on the field. So when we consider the net soil greenhouse gas emission, accounting the N2O and the soil carbon stocks, it's possible to observe that the carbon stocks change were more relevant in terms of net soil GHG emissions. And only in the TR scenario, total removal scenario, the soil acts as a source of greenhouse gas emission. And in the other scenario, where at least one part of the straw was maintained on the field, the soil acts as a sink of N2O. this especially because the emissions, the accumulation of carbon associated with the maintenance of the straw on the field. Here we can see the bars associated with soil carbon storage, and here we can see the net emissions. So as a conclusions of my presentation, I would like to mention that the straw removal reduces the effect of the soil as a sink of greenhouse gas. And we advocate that the effect of the straw removal on soil greenhouse gas balance should be considered in studies that evaluate the benefits of sugarcane, the sugarcane straw-based bioenergy. So we advocate that these values should be included in future LCA of the bioenergy produced based on sugarcane straw removal. So I'm finished here. I would like to thanks to everybody and goodbye.